Good afternoon and welcome everyone. Hi, I'm Cheryl Reynolds and I'm with the UC statewide IPM program. Welcome to the UC Ag Expert Talk on Laurel Wilt. Peter Cosina is here with me and he's going to be running the polls today and troubleshooting technical problems. And now I'd like to introduce our speaker today. Dr. Monique Rivera is a cooperative extension specialist in subtropical entomology at UC Riverside. Today she's speaking on the Laurel Wilt in avocado. And now I'll pass this over to Monique. And so Monique, you can go ahead and share your slides.
Okay, so I'm going to give you a brief outline of what I'm going to go over today. And I'm hoping you guys can hear me okay when I'm not muted. Okay, so I'm going to run through an introduction, a somewhat brief introduction to the Laurel Wilt pathosystem, and then discuss the vector biology and ecology of ambrosia beetles, the current known location of the disease within the United States, tips for identifying the disease, and what we think are the best practices for prevention and management if it potentially makes it to California. Okay, so when it comes to the Laurel Wilt pathosystem, I first want to define for you what I mean by pathosystem. That's kind of a jargon word, but it's really helpful to be able to talk about the all-encompassing factors that contribute to this disease pest pressure. So basically, you have a dynamic of many different things that center basically on the vector. This is a vector-borne disease, so we are concerned with managing the vector so that the vector doesn't make contact in order to spread the disease. So this includes, when it comes to the pathogen, the population size of the vector can influence the population size of the pathogen that's present, as well as the virulence. And there'll be follow-up slides that explain a little bit more about what I mean by that. The plant host, they vary in their levels of susceptibility. Okay, I'm seeing, hold on, I just want to make sure that I don't have any comments. It's very echoey. Okay, well, I don't know how much I can do about that. All right, so all of this basically occurs within an environment, and the environment's made up of biological and physical characteristics. Let's see. So again, like I said, this is a vector-borne disease, and all of these different elements make up a really complex pathosystem, and it's one of the most difficult to work with outside of probably citrus greening, where they can't actually culture the pathogen. So in going over some of the pathogen details, so Rafaela lauricola, that is the fungal name of the fungus that causes the Laurel Wilt symptoms and gets into the vascular tissue. It's also called an ambrosia fungus. That's because it's linked to ambrosia beetles. The fungus itself is what causes the Laurel Wilt. So it's nothing to do with the beetles and their development on the tree. It is specifically due to the pathogen. And the beetles need the pathogen in order to consume the tree. It gets its name, laurel, from Lauriaceae. That's the family of trees in which the pathogen attacks. And then wilt, because it has to do with the fact that you see this wilting characteristic to the leaves to begin with. And that's because the fungus affects the plant's vascular tissue. And so vascular tissue can be somewhat of a jargon word if you're not familiar with it. So that's why I have these slides here. So this celery up top, you can see along the bottom there, those little circles. Those are their vascular bundles. And I think the key idea here is, or the way I like to relate to this idea is that it's basically veins, but it's plant veins. And that's why this picture in the bottom where the celery is in different colored fluids, you can see the absorption of the color into the leaves, because that's how the color is making it up into the plant, that vascular tissue. So the plant host is the whole family of Lauriaceae that we know that exists in the United States. This includes avocado, of course, but also bay laurel, which is, laurel, the bay leaves that you use for cooking, the trees that's collected from. California bay, which is native to California, so obviously we want to try to protect that. And then camphor trees, which are also somewhat present in the landscape. So in terms of the environment, so right now the disease is spreading throughout southern Florida in avocado, but obviously California is vastly different, and there's a couple things that really make this difference significant. So one is humidity. We have much less humidity here in Southern California than they do in Florida. And I can attest to that because I came here from Florida last July, and it's almost a complete opposite humidity issue here. So rainfall is very different. Florida is more subscribing to a tropical nature where, especially in southern Florida, where they grow avocados down around the Miami area, they're pretty much having a rain every single day around 3 to 4 p.m. So the elevation and terrain, Florida is extremely flat and is at sea level. So throughout Southern California, you have a lot of different variations going on with elevation. And then that leads me to my next comment, which is avocado's integration into the landscape. So in Southern California, I've seen avocados be placed on the side of a hill, and they can be very close to housing developments. In Florida, it's somewhat similar, but I would say that the area that they grow avocados in is mostly more rural. It's outside of Miami towards the Homestead area, where's the bulk of their acreage. So like I showed in the pathosystem slide, there's kind of a box around it because this entire environment influences every aspect of the pathosystem, and in particular, the vector and the vector's survivability. So the vector is ambrosia beetles, and you might be looking for me to cite out a certain species, but I don't want to do that because, and I'll explain a little bit about why, recent findings. But basically ambrosia beetles are incredibly tiny. You can see on the penny here, that's for size, on average, about how large they are. The red ambrosia beetle is the one that's been classically associated with Laurel Wilt, Xyloborus glabratus, and it is considered the primary vector in forest stands of Lauriaceae. We haven't actually found this in avocado. If they found it, it might have been 1 or 2 beetles, but it's not considered to be a part of the population in avocado. And so a key thing to know about ambrosia beetles is they're fungus farmers, and they hold the spores in a structural pocket called mycangia. The mycangia is circled in red in the top right of the photos. It's basically just a little divot pocket in their exoskeleton that contains all the different spores of the various fungi that they carry with them. And so like I mentioned, Xyloborus glabratus, not found in avocado. I was just extensively sampling and identifying last summer in avocado, and we found 0 Xyloborus glabratus, and it's very unclear what the specific vector is, and that might have to do with the biology. They may have jumped too soon to conclusion that it was one specific species. There seems to be lateral transfer of the pathogen between species potentially. So this slide gives you a bit of an overview of their life cycle. So if we start with this tree trunk here with the 2 red arrows, first you have female beetles emerging from whatever host they developed in initially to search for more host trees. So with almost all the species, they have these foundress females that, depending on their life history strategy, meaning how they mate, what they need to produce their next generation, it really depends on what that life history strategy is. But in many cases, you can have a single foundress female that can completely just lay eggs and start a population within a tree. So in the second picture here, you can see the spore mycangia here. And then depending on what species you have, and this is where ambrosia beetles get complicated, but that's sort of what I'm trying to convey in this talk. is that this is more complicated than simply a single species of ambrosia beetle with one single fungal pathogen. But depending on what fungal pathogen that they have, and if they do have Raphael loricola, that particular pathogen breaks down healthy plant material. So the ability of that pathogen is what allows it to break down healthy trees. Normally, in the ecosystem, ambrosia beetles mostly function to break down dead and dying trees. Well, this is unfortunate because this kind of starts the process of that, and you can attract larger populations of ambrosia beetles if you have this foundry population that infects the healthy trees. So basically, they'll bore into the tree, much like Polyphagus shot-hole borer, and leave those little matchsticks, wooden matchsticks looking sawdust coming from the outside. The beetle then releases its fungal spores into the sapwood, and that's what initially, they're basically starting their farm. They're going to put those spores there, let the spores break down the plant material, and then feed on the fungal deposits. And that's when you start to see this crown wilt symptomatic feature that goes on in the next portion here. You'll just see a wilting behavior and a discoloration of the leaves. Also, if you take the bark off the tree, you'll see a mild discoloration. I have photos of that later. So when the tree is completely dead, you'll have a dead and dying upper canopy, and then you'll also have the tubes that are coming out of the tree as the insects exit wherever they want to. And then once they're dead and they exit, I mean, when you open up the tree, you can really see the galleries, and I have pictures of this later on in the talk as well. But it can be quite loaded with beetles, and I think from a management perspective, it's quite difficult because you may have seen the frass tubes, you might have the wilt, but until you get in there, I mean, in order to identify what species you have, you have to collect the beetle populations, and this takes a technical skill to ID these beetles, either that or molecular tools. And in the slide, or in the picture that's next to the life cycle, you can see different stages of the laurel wilt. So the early stage is just a coloring at the tips of the new leaf material. And then once you get to the intermediate stage, you'll see whole branches that have that wilting discoloration feature. And then at the late stage, when you have a total infection, everything will be discolored and the tree will be basically on its last legs and dying. So I know I said Xyloborus glabratus is not necessarily the vector, but it is sort of the vector that generated our knowledge of this particular pathosystem. And so it's important to just understand that while this might not be the main vector in avocado, it is the guiding species that they find mostly in natural stands of Lauriaceae, and they do believe that in natural Lauriaceae, it is the actual vector. And it was brought in from East Asia, and it's absolutely strikingly changing the native composition of forests in the southeastern US. It's wiped out 500 million Lauriaceae trees. And that includes these species here, red bay, silk bay, sassafras, and swamp bay, which made up a large portion of the forest there. So I think with that, we'll have our first set of questions, if Peter wants to put them up.
Yes, so he just put that first one up there. Okay, and you should be able to see the results screen now.
Great, so most people seem to have answered that correctly. There's also somebody who decided that the environment was the main thing. That's not 100% wrong. The environment contains everything. But yes, the correct answer there was all of the above.
Okay, so now we'll bring up the next poll. Okay, so now that one's going to be closed out and we'll see the results.
Awesome. We have the vast majority of people answering true. And so that's a good sign. So we can move on to the next question.
Okay, and that one should be showing up. right now. Okay, we'll be closing this one out and bringing up the results screen.
Okay, great. So we have most people answering those questions correctly, which means that, you know, these main messages are getting across, and we can continue with the webinar. So now I'm going to get more into the biology and ecology of these ambrosia beetles. I know it now seems ominous because we don't have a specific species that we can point our fingers at and say, yeah, that's the one. But that is kind of the way that we need to be thinking about this. So it's good ultimately. So ambrosia beetles can invade healthy, stressed, dead, and dying trees. Again, their ability to infect a tree is more so guided by the fungus that they're carrying with them. So it's almost like they need that fungus in order to eat, and what they can eat is dictated by the fungus and what the fungus can successfully infect. And so, like I said before, the primary ecological function of them, when the fungus is not a plant pathogen, is to degrade dead and dying trees. So they are these, you know, post-tree death or when the tree is almost dead, invade the trees. Now, what becomes a bit of an issue is that when you have all these different abilities to infect the tree, this is where the most current research thinks that what could be happening is lateral transfer. So what I mean by that is that you have one beetle here that's feeding on Raphel, and then you have another beetle here, and if their tunnels overlap, there's a chance that these other beetles that are not whatever the carrier species or the initial infection species is, could be picking up that plant pathogenic fungi and also becoming vectors. So it's a dynamic thing, and I think the approach in Florida is now becoming more towards population management rather than looking for specific species. But the one thing I want to emphasize is that they don't directly feed on the tree. I think you guys got that based on the last questions, but it's important to know that the beetles are just the carrier for the fungus, and they need the fungus in order to feed on the tree. So that is key when you're thinking about disease management. If you have the beetles, it's likely, if you find that population of beetles there, it's likely that they've spread some amount of fungus, but we don't know specifically which one it is because the associations are promiscuous, which brings us to the next slide. So this figure here beautifully describes how complex the diversity of these different species are. And just to give you an example, so we're finishing up a manuscript from my fieldwork last summer in Florida with Laurel Wilt, and we were finding upwards of 20 species in a field, depending on what attractant or repellent we were using. So you can have a really high diversity of beetles, but what you actually want is no beetles at all. And I'm just repeating yet again that the ambrosia beetles are only as strong as their fungal symbionts. So if they are carrying, they happen to be carrying the Laurel Wilt pathogen, then that means that they have the ability to potentially release that pathogen into a healthy tree. And this pathogen is, it's not only able to infect healthy trees, but we're talking about really quick tree decline. Within a matter of a year or two, you'll see the symptoms and the decline of the tree happen really quickly. And like I've said before, there's a bit of a, what they're calling a promiscuous association. So for example, if you take another pathosystem, the citrus greening HLB pathosystem, you have citrusillid, and they're specifically carrying the Libiribacter asiaticus or some other type of Libiribacter, and that's a really tight linkage that seems the most up-to-date evolutionary theory I've heard on that would be that it's basically a symbiont gone rogue in that it's no longer necessarily beneficial. It's not a digestive beneficial, it's just riding along and infecting as much as it possibly can. So what I want to emphasize though is that there's not a strong linkage between any one species and the specific fungi that it carries, in the sense that every species carries its own little pocket of diverse fungi, and it just matters what's in there. So they haven't done enough basic research to truly know what guides the linkage or what allows certain species to carry it and maybe not others. I think that if you're following the literature on this, what I've noticed is that all the time they're discovering new species that they're finding the Raphael loricola in, and so it's probably pretty likely that there is this lateral transfer going on. Now they're running studies on what beetle species can actually eat that fungi, and if they're eating it, then it should potentially be the primary fungi that they carry with them. So just to emphasize again, presence of the beetle does not always suggest presence of a specific symbiont. So if you find Xyloborus glabratus, will it be carrying Raphael loricola? Maybe, but that becomes a very molecular question. You'd have to take the beetle in and have it tested. So then the question is, what is going on in avocado? We don't find Xyloborus glabratus there. I was collecting traps every week for about 6 months, and we never found Xyloborus glabratus in any of our traps, and this has been previously reported in the literature as well, that they don't find that specific species in avocado. The species complex, in fact, is different from what they find in forest. It's not necessarily more diverse, but if you were to plot them out on a map, you would see many different species in avocado versus in the forests. And what we know from the most up-to-date research is that multiple species can carry Raphael loricola. Again, it's hard to define them as pathogens if you're not specifically linking them to the infection in the field. So this is why this has become an ambrosia beetle population management project rather than we need to keep this specific beetle out. So this last bullet point here really refers to my lateral transfer thing that I'm commenting on. It's that if you have that particular fungi in a tree and it's there first, it's possible that other species that are invading that already dead, diseased, dying tree are then picking up the fungi and also reproducing and moving it to other trees. So in avocado, really the approach, and I've already repeated this a couple of times, but it's important to emphasize, is to control the population of ambrosia beetles. So we're trying to redesign the way we're thinking about this and not just looking for one species, but trying to keep them all out. And like I previously said, a single female can initiate the development of a local population. So you have a single female can have upwards of 20 beetles in her, I guess, clutch of beetles that will then move on to different trees. So it's an exponential growth. However, I did see success in Florida. It's not 100% hopeless, but it's a very intensely managed situation to keep the avocado disease-free. And it really always comes down to conscious management, which means scouting and tree removal. So just making sure that you're aware of your particular grove and what your action plan is. I mean, that's what I would encourage, is like, if Laurel Wilt is to come, my action plan will be, that is probably going to be the best approach at this point. So right now, this map basically shows its entry point, which was in 2002, and this is, you know, where we linked Xyloborus glabratus specifically to being the vector in the natural stands. So that's basically the southern tip of South Carolina. And since then, you can see these striped counties over to the left. And those are the newest points that they have found them in forest stands. So you can see that it's been gradually moving along the southeast since its arrival. Quite quickly throughout this eastern seaboard area, the base of Georgia and top of Florida. And then the growing areas for avocado are basically this 2011 on the tip, bottom purple area, and then all along this orange to the left of it section. So it's been there, and they've actually predicted out, modeled out about how quickly it would be spreading along this Gulf Coast. So this is a pretty conservative estimate. I think they're using this, they're modeling this based on how they've seen it spread currently. However, if you have every guy and his brother, sister, mother-in-law moving around firewood, then it's going to move much quicker. So there's been a lot of campaigning around not moving firewood. We're just not exactly sure how effective it is. And again, it takes, you know, one log moved with a population of ambrosia beetles that, you know, you can establish a foundry population in a completely new area and have an invasive pathosystem on your hands. And so that's why this messaging is critical. And the more we can promote this idea of not moving around wood and wood products, or making sure that it's treated, is going to be really the key to preventing this from coming to California. So with that, I will let Peter go ahead with our questions.
And while our participants are answering this question, Monique, we did get a question in the Q&A for you.
Sure.
So this one is, so what implications does this beetle and its fungus have for spread to California? And then also mixing with the Polyphagus shothole borer and its fungus.
Well, the Polyphagus shothole borer, I would say, is somewhat less scary than ambrosia beetle, just because of how they've seen that disease completely spread in Florida. Again, I think that this all hinges on not moving wood and wood products around, because otherwise the beetles would have to, they're not very great flyers and they can't fly very long distances. So I would say that it making it across the desert on its own, not going to happen. But somebody with a log with the insects in there making it across the desert, that's more probable. But the implications overall, I mean, I'm not as familiar with the Polyphagus shothole borer research as I am with the Laurel Wilt ambrosia beetle research. But my understanding of that, at least since I've been in California, is that it's become less of a concern for avocado growers as it's moved into its preferential hosts that are actually in urban environments. But someone can correct me if I'm wrong on that.
Can the beetle be moved with fruit from Florida?
No, they have no interest in the fruit. And in fact, when they've done studies on the preferences of ambrosia beetles between the different Lauriaceae species, avocado is among the least preferred. So I would think that it would move along the forests. I think that the most at risk, obviously, is going to be the southeast and throughout, where they have those natural stands. But moving wood around will move the beetle around as well. So the answer to the question that I posted is definitely movement of wood. Grafting of infected material, I think that based on our awareness of the various diseases that can be spread, I mean, maybe, but I kind of doubt it, because the tree that has the Laurel Wilt will look so bad that no one's going to want to graft it. And then fungal spores from Florida, not going to happen. They're not going to move without the beetle. So we can go to the next one.
Okay, so that second poll should be showing up. And if anybody does have other questions, feel free to put those in the Q&A and we'll pass those on to Monique. And it looks like we do have one. What is the cause of the wilt, and do spores move into the vascular system?
So the cause of the wilt is specifically the fungus. And it's the fungus moving into the xylem of the tree. And it's not necessarily the spores that are moving, but it's basically killing off the plant material. So it's killing off, I'm trying to think of an easy way to explain that. It would be like if you were drinking out of a straw and then all of a sudden somebody cut it in two. It would just, or smashed it in the middle and ripped it and put holes in there. It's just messing with the transfer of the veins, which is causing the wilt in specific locations. And so, of course, you know, what some people might do is they might be like, oh, well, I see the wilting, I'm just going to cut that off. Once you see those wilting symptoms, it's already too far gone. The beetle's there, the fungus is there. So pruning isn't really an option. That's when the strongest recommendation I've heard in that situation is just to go ahead and find out, make sure that the tree is positive and remove it. I hope I answered that question correctly. So looking at our poll results, most people did get that we don't know the conclusive answer to this question. I'm glad to see that. There's still Xyloborus glabratus, the red bay ambrosia beetle, coming up with 8 people answering that way. What I would say about that is that's not necessarily, as long as you're aware of the disease and you have the concepts down, it's okay to say that it's Xyloborus glabratus. I'm just trying to make sure people are updated to the most current way that they're actually thinking about this in Florida. So it is true that we don't know the conclusive answer to this question. I think we have one more, right?
Yes, there's one more. And I do have another question for you. How many different fungi have been found in ambrosia beetles in Florida and its infected trees?
That's a really hard question for me to answer. I know that there's been that those mycangia host a diversity of different species of fungi, and that their quantities of the species composition, if you just imagine it like, you know, a pie bar graph, are different between even individuals that they've extracted. But with them being contained, I don't know that they've really looked for, in particular, other funguses that are successfully pathogenic within the tree. They're mostly looking for the laurel wilt because they know that that's like the front causal agent. That's the thing that gets into the tree that starts the killing process. And then once you have a bunch of other species of ambrosia beetles trying to capitalize on the dead and dying tree, those aren't really pathogenic per se. They're just there to break down the plant material so that the beetles can eat the tree, the dead and dying tree, to gain the nutrients. I hope I answered that question appropriately. Please just follow up if you need more information. Okay, so this is great. Most people are getting that the messaging on the poll, 79% are understanding that the ambrosia beetles are feeding on the fungus. They're definitely not feeding on the trees. They need the fungus in order to feed on the trees. And that's a concept I'll be continuing to push throughout the talk. Okay, so I'll try to make it through this last section, and then I will be around to answer all the questions and comments coming in through the Q&A. So I've shown this before, an aerial view of the disease progression. So once the disease is showing up and you see these symptoms, it's going to be important to be aware of them. And we'll have to come up with a best management practices probably for California to figure out how we can help growers know what's going on, how to get their plant material tested. That's something that UCIPM will have to come up with for the website. So this is a picture from Florida, and this is a natural stand, and you can see this extreme dieback of branches. I did want to mention, so another thing that was buzzed about that they were trying to figure out in Florida was they think that this fungal pathogen has the potential to root graft onto the tree next to it. That was under investigation, but that was something that they were seeing basically potentially coming up with the disease. The way it was showing up in fields was making them think that that was a potential way that it was spreading as well. And because in avocado, of course, you have all the same species of tree growing, that could have been contributing greatly to the spread within the avocado groves. So this is what the inside of the sapwood looks like when you have a beetle population there. So first of all, you have this staining that looks just like a wet spot. And that's basically the failure of the xylem tissue that leaking out of materials that was in there. It's a staining that you'll see underneath the bark. And then also, of course, the galleries of the beetles you'll see within the trunk there. And then this is a characteristic sign of any sort of ambrosia beetle, these what they call matchsticks, which are the sawdust that protrude out from the tree when the beetle actually enters. So when it comes to identifying the disease, this is a list I made up of what I think is a visual symptom summary. So first, the wilting of the leaves and young stems. So it will primarily happen in the youngest tissues, and that's because the plant is trying to move nutrients to those places so that they can grow. And what that means is that those are not getting enough of the nutrients because the vascular tissue has been disturbed. And then secondarily, the leaf color change as it lacks nutrients. You'll see dark purplish green and greenish brown, and then eventually dead leaves hanging on the tree. The woody material will also start to die back. So stems and limbs you'll see individually will go first by way of wilting and then be completely dead. There will also be dried sap on the trunk. So upon inspection of the trunk and any major limbs, it may show dried sap, and that's basically, I think, the tree trying to react to its own stressor. And then, like I just showed a couple slides ago, the streaking of the sapwood, that basically like wet look below the bark. It's a staining. And it also may be streaky looking as well, depending on how far the disease has progressed. So there will also be small dark holes in the sapwood, and that just indicates that the wood-borne beetles are present. So like with any vector disease, I just want to point out that just because you have the visual symptoms does not mean you have Laurel wilt. You will need to have that confirmed via molecular techniques. So currently in California, we're in prevention mode, and I think what that means is, again, stopping the movement of wood and untreated wood products from the region of infestation into California. If we could stop that, our chances would be much lower. And then, of course, monitoring, scouting. I like to tell all growers that I talk to, know your grove, in the sense that your visual understanding of what's going on is always going to guide our sort of first alerts about what's actually going on. And that will help us get early detection of any potential infections. So how might one monitor for this disease? Again, scout and know your groves, keep track of potential symptoms and evidence. Stay in touch with updates on the disease. If they were to find this, there would obviously be a huge blast alert to let everybody know that this had made it to California. And then in development, there is an ethanol lure and a white sticky trap. In particular, if you know there are ambrosia beetles in your area, this might be a good way to just see what's there. But again, you'll need experts to identify what in particular is there. So this is an ongoing tool. We used this in Florida right before I left in a study. So hopefully this will make it to market and be something that can be used as basically something to know what your population looks like. But then again, you're going to have to work with experts. But if you can get to the point where you can identify what an ambrosia beetle in general looks like, it might be good, but we would need to do more training on that. The ethanol lure is, we call it a low dose ethanol lure. And basically what this is it's bringing the ambrosia beetles in because they think there is a dead or dying tree. So how might you know for sure if it's Laurel wilt? Well, the disease should be confirmed by molecular tools. In particular, PCR will confirm that it is indeed fungus. And if you have something that looks like it might be an infection, get your tree sample and confirm it's Laurel wilt before removing it. It's a disease we're going to establish here. You could go more so on the symptoms, but this is a very early prevention mode attitude. Also reach out to your local extension specialist or researchers get in touch with somebody about how to get the material tested. I'm not sure whether California is specifically set up for this yet, but I think that our fingers on the pulse and that there will be something very quickly if the disease should arrive here. So I also wanted to include a little bit about previous work that they've done that I think is relevant to California in particular. So they basically infected a bunch of different varieties, and that's shown here in this table, with Laurel Wilt to understand how disease severity varied by variety. So it's important to note that when I report on the varieties I think are most important to California, that the overall average for their rating of disease severity, which was from 1 to 10, the overall average of everyone that was tested was 3.7. And then the maximum that they saw in terms of disease severity after 3 years was 5.8. So hops, which is obviously very important to California, it was rating at 2.7, bacon at 2.1, Pinkerton at 3.3, Reed at 3.5. They did not test Forte and Gwen. So I think the implication for that is that the disease symptoms seem to develop faster in Pinkerton and Reed. But by how much? This is on a relative scale. So I'm not sure. And these are also greenhouse grown and held in the greenhouse to be able to do this. So I don't know how specifically applicable it will be into the field, but it's a great lead-in and indicator of what varieties might be the most susceptible should it arrive. So what is going on in Florida? They have this, it's obviously killing off a lot of trees, natural and otherwise. And basically when I was there, there were growers that were successfully managing the presence of the disease and ripping out trees and replacing trees. But this was all hinged on using an integrated approach to save as many of the trees as they could. And that really involves removing the trees and then chipping the wood to kill off the beetles. So if you go out there, you remove your tree and you turn it into logs, well, the beetles are still living in the logs, so it's not good. They have to be chipped, which is unfortunate because that makes the tree removal that much more expensive. They also treat the surrounding trees after removal with either insecticide or fungicide, depending on what the manager has in their arsenal and how they're dealing with it. They limit or time pruning. So again, the beetles are attracted to dead and dying trees. Regardless, the whole population will be attracted to dead and dying trees. So they basically try to time their pruning with their insecticide and fungicide applications because they know that the pruning damage causes this plant material to be more attractive. And then what we were working on before I left Florida was behavioral manipulation of the total ambrosia beetle population. And we were trying to develop a tool and hopefully it'll become commercially available at some point in the near future. But we were basically using a push-pull strategy, meaning that we were using our ethanol lures to pull the beetles in and trap them on traps. And then as well, we were using a repellent to repel them from individual trees that we were trying to protect. There also are potential biological control agents that are under investigation, such as predators, parasites, and entomopathogenic fungi. So this is always going to be an interesting, it's always interesting when the biological control agents get introduced because I think that it all depends on how effective they will be, on how much specialists and advisors can recommend their use. Because when you're risking the complete plant death with the disease, it's hard to recommend something that's only 60% effective. I mean, if you're going to lose the entire tree, then maybe that's not the best particular approach. However, I do want to say that these things can be very effective, but the jury is out for now on what they will come up with. So a lot of times I think when you have this type of situation where you're basically asking people to remove trees, it's also important to explain why that's being recommended. And that primarily hinges on the fact that we're not sure if root grafting of the fungus occurs between trees. They're currently doing experiments, but it's a very difficult thing to investigate. As you can probably imagine, you have an established grove that's been there for 5, 10, 15 years. how are you to know whether this is being grafted? So this is, you know, greenhouse experiments, et cetera, and we're hoping to hear more back about that in the coming years. And then additionally, each tree can have a large and diverse beetle population within it, and that promotes the access and spread of the fungus. So if you remove the tree and you chip it up, again, the key thing is they're chipping up the tree, you can actually get rid of a portion of your beetle population with that action. And so with that, that is the end of what I had to present today, and I hope that I left enough time to get everybody's questions answered. I know we have 3 or 4 poll questions, so maybe we can ask in the interim while the poll's up.
Yeah, so that first poll is up, and while people are answering this one, I can ask one of the questions we received in chat. So when ambrosia beetles are tunneling in a tree, are they just removing the wood tissue to create their tunnels and not actually eating the wood?
Yes. So those little matchsticks that come out are just them. Basically, imagine if you're digging a hole in the sand and like the excess sand that's coming out of the hole, that's what's going on there. So they're not eating the tree at that. They're never directly eating the plant tissue. They're always eating their fungus and the fungal byproducts of the tree that are being broken down by the fungus.
Okay, so, and we had clarification on that earlier question. So this one is, does the fungus produce cell wall degrading enzymes such as cellulases or xylanases?
I don't know, I could look that up for you, but I would say that that is pretty standard for any plant pathogenic bacteria or fungi to have some sort of enzyme breakdown for the plant material. That's of course also the reaction that's feeding the beetles. So I don't know specifically what enzymatic reaction, but I do know that that's very likely the, I guess, methods of how they're breaking down the tissue. And I see the poll results right now, and the vast majority of people are answering correctly. And this is one that I didn't specifically talk too much on, but it's very interesting to see people's answers. So the answer was all of the above, and 77% of people answered that. Broadcast sprays don't suppress the populations because the populations are within the tree. And then if you already have destruction of the vascular tissue, having a systemically transmitted through the tree insecticide is not necessarily going to be effective at getting the insecticide to the beetles. So this is why this disease is in particular really hard to manage. So you can go to the next one, Cheryl.
Okay, and that one should be up. Okay, so we're going to be bringing up the results for this poll.
Okay, great. So all the above was the answer there. So there are potential things out there that people are investigating, including here at UCR. They're also looking at entomopathogenic nematodes. They're actually importing material from South Korea to look at the different biological control agents. Of course, this is all under high quarantine, so no worries there. But we can move on to the next question.
Okay, and just a quick question. Is anyone trapping for this in California?
That I'm not sure about. I know that I'm sure APHIS, the government agency, is very aware and keyed in and is likely doing something. But if there is something being done in California, I'm not specifically familiar with it.
Okay, I think we're going to close this one out and bring up the last poll, or the results for this one, then we'll bring up the last poll.
Okay, great. So most people understand that the detection is hinged on molecular methods, which would be the PCR polymerase chain reaction. Dead tree branches and stains on the bark are both visual symptoms, and while they are strong indicators of the disease, they do not confirm its detection. So we can move to the next one. And I think this is the last one, correct?
Yeah, this is the last one. And then one question during this one, do matchsticks occur with other ambrosia beetle feeding?
Yes, it's a really common symptom, including with polyphagous shot-hole borer. That's why, you know, we don't want to depend on the visual symptoms for this.
And I bring up another one here. How do the repellents work that push ambrosia beetles away from highly valued trees?
So it's actually a technology that's already available. It's a technology by ISCA. ISCA is a company here in Riverside, California, and the product is called Splat. So all they basically need to do is make the splat with the compound that we were using commercially available, and then it's able to be applied. Splat is a flowable matrix that allows compounds to volatilize from the matrix. And so basically, the technical term for this would be semiochemical because it's communicating something to the beetles. But yeah, I think that hopefully that's commercially available within the next year or so. However, applying Splat can be kind of tricky. I know that you can do it with tractors and stuff, but we're still in the process of investigating specifically how that should be used, in the sense that it has a less than a meter range of efficacy. So it means that it needs to be applied much heavier than we originally thought that it would be. So the best management strategies currently available, I can see our results. So I would say that this is interesting because most people are saying removal of infected trees, but the actual correct answer there is all of the above. So I want to emphasize that we do want to use an integrated strategy. The trunk-injected fungicides, I saw them being used in conjunction with other strategies quite successfully. But removal of infected trees is an unfortunate truth. And a lot of times I would go into groves in Florida and you would just have so many of the trees stumped from this. So I don't want to see that in California, but I'm glad that people are understanding the seriousness and how removing the trees is really this is something that needs to be taken seriously. So
I did have a follow-up to the splat you just mentioned. Mm-hmm. And so that one is, so the AI is mixed into the splat and it volatilizes, or does splat have the AI already in it?
So when you purchase the splat, the AI will already be mixed into it.
Okay. And so there's a question, if you were to guess where this would first turn up in California, where do you think it would be?
Somewhere in Southern California, I would assume. That's really hard to say though, because again, when you have all of these people and the potential for the movement of wood, if they have the right alternate host there to move from the wood into a natural tree stand, then that's all it really takes.
Okay, and then there's a question from Brad. Will the granulate ambrosia beetle transmit Laurel Wilt? And then he says, I guess so, as lateral transfer may mean many or any ambrosia beetles can carry a range of fungal symbiotes. Yeah, so basically, I think that the approach right now and the rate that they're finding out different species can actually carry the fungus, the best approach is to assume that, you know, the ambrosia beetles that you find more than likely have this pathogen or something else. But again, like getting everything tested if you find ambrosia beetles is also really will help understand what's going on. I think our key thing here is that we just don't want to establish an endemic population of these beetles, and then from there it's manageable and preventable. Okay, and then there's a question, would thermotherapy help reduce ambrosia beetle population within the tree and slow down the disease progression?
It's an interesting idea. I don't think that they've tried that, but it would be interesting to know more about that. So my impression of thermotherapy and my background in knowing about its application comes more from citrus. And it seems like thermotherapy, why it can help reduce maybe the so-called quantity of the fungus, it's probably not going to erase it completely. And if the beetle is helping promote its spread within the tree, I think that there's a chance that the beetles could just re-inoculate the tree after the thermotherapy. I don't know how hot you'd have to make it in order to kill off the actual beetles though, but it's a very interesting question. I just think that the disease progresses so quickly that growers in Florida are just, it's a more viable solution for them to take the tree out.
Okay, and then there's another question. Won't chipping wood still allow the tiny beetle to disperse from the site? Is burning felled material on site the real only method of killing the beetle?
So they've actually shown conclusively in research that the chipping does actually take out the beetles. I think that you're right to ask, because I guess it depends on the size of the chipping, right? Well, I'm thinking like as fine as you can get it. They're still going to need the plant material to eat off of. But I think that they've shown fairly conclusively, and any question that you have that I'm not necessarily emailing or I'm not answering very well right now, please email me and I can give you specific references to studies and other things for you to read. But yeah, I think that they've shown pretty conclusively that wood chipping is actually really effective, and I do have, I have read that, so I know that there is data on that.
Okay, so a couple more questions. Could fungicides be used to prevent or disrupt the food source?
Yes. The fungicides are really expensive, so using them prophylactically is going to be, you know, that is up to grower discretion, how much they want to invest in the prevention. But I've seen the trunk-injected fungicides work very well. So the grove where I was doing a field study was right across from, it was basically a tractor row away from a field that was managed by another grower, where they weren't really removing trees and they were basically not managing it, versus across the road you had fungicide management, repellents, very quick reaction to removing trees, and those trees looked like they were intact minus the gaps where trees had been removed. So I do think fungicides are really a viable thing. We'd have to look at prophylactically at how effectively it would keep them out, keep the disease out of the tree.
And just a couple more. How much do secondary pathogens contribute to the death of ambrosia-infested trees?
That I'm not particularly sure about, and I think that the pathogens that would affect the trees here would be different from what they were seeing in Florida. I think that it's probably a standard schematic to know that anytime the weakened tree is present, the secondary pathogens are going to increase the decline really quickly, but we would need to do research here in California to understand more about how our specific community of diseases would interact to influence tree decline.
And I think this is the last question. Are there any entomopathogenic nematodes that can be sprayed on trees, enter tunnels, and attack the ambrosia beetle population?
So they're looking for entomopathogenic nematodes that are coming from the logs that are being sent to quarantine here at UCR. They're looking for entomopathogenic nematodes. So far, they haven't been able to confirm that any nematode that they've extracted is actually killing the ambrosia beetles. So this is something that's in motion. Right now, there are no available tools, as far as I know, in terms of entomopathogenic nematodes. But then on the other hand, I'm not particularly sure how much work has been done with the currently commercially available species.
Okay, so that is all of the questions we have. So thank you, Monique, for taking the time and presenting today.
And please feel free to email me. I'm here to answer your questions even beyond the webinar.
And thank you, and have a good rest of your day.